Hello, hi, a very happy good evening. So this is Krishna Ma'am. We're talking about the story of life on earth, and that's biology. So thank you so much for joining my new beginning series about human reproduction. So please don't forget to like, share and subscribe. So we are going to continue our lecture about gametogenesis. So today I hope I finish off with gametogenesis, but let's see, because we have a bit of portion pending in your spermatogenesis itself. So we are supposed to talk about the structure of the human sperm, and only then we'll be able to move forward to your oogenesis. All right, yes. So here we go. So because spermatogenesis is important, so I have taught you the process of spermatogenesis. I'll give you a quick recap, and then we'll move into the process of your sperm formation, the structure of your sperm, and then we'll move to oogenesis. So I hope I can finish, but let's try our best. All right. So here we are. So human reproduction, lecture 6 or part 6, you can say. So yeah, so we are talking about gametogenesis here. So oogenesis and your menstrual cycle are the two huge topics. So rest everything will be done in 1 or 2 lectures. So don't worry, it will not extend to much more or much longer. So we were talking about the process of gametogenesis. So gametogenesis is the process of formation of gametes. So genesis in Greek means formation. So we have 2 types of gametogenesis. One is spermatogenesis. The other one is oogenesis. So spermatogenesis is the formation of sperms. Oogenesis is the formation of the ovum, that is the egg. So in the last class, we were talking about how spermatogenesis happens. Excuse me. Yeah. So we'll continue with spermatogenesis. So the process of spermatogenesis begins at the stage of puberty. So we know in male the primary sex organ are the testes. So inside the testes you have compartments which is called as the testicular lobules. So which in turn has 1 or 2 highly coiled structure that is called as your seminiferous tubules. So this seminiferous tubules actually have 2 types of cell. One is your male germ cell. So this male germ cell is also known as the spermatogonia. So spermatogonia is plural. So if I write spermatogonium, it is singular. Right? And it also has another type of cells that is called as the Sertoli cells. The Sertoli cells basically nourish the sperm cells. So these are the sperm cells. So I can write they nourish the germ cells. So we are going to talk about this cell, the spermatogonia cells. So what happens? So let's see. So this group of spermatogonia, first they undergo mitotic division. So mitotic division is equational division. So where the chromosomal number is the same, right? So they undergo mitotic division to increase their number. So now you have increased number of spermatogonia. So among these increased number of spermatogonia, so some will be willing to undergo meiosis process, some will remain as stem cell. The one which is willing to undergo the meiotic division is called as the primary spermatocytes. The one which is willing to undergo meiotic division are called as primary spermatocytes. So here you have your primary spermatocytes. So primary spermatocytes undergo meiotic division 1. Okay, so they form 2 secondary spermatocytes. So meiotic division 1 is reductional division. So primary spermatocytes are diploid, so the secondary spermatocytes are haploid, okay. So this is your haploid one. So these two are your secondary spermatocytes. Clear? So far you're clear? You guys are able to recollect what we were discussing in the previous class? Yes? Shall we continue? Okay. So these secondary spermatocytes, each of them will undergo second meiotic division. So meiotic division 2 is similar to mitosis, so it is equational division. Right? So this is equational division. So now each of them will form two haploid cells. So these are called as spermatids. Excuse me. So one primary spermatocyte divides to give you 4 spermatids, because at the end of meiotic division, you know you have to get 4 daughter cells, right? So when we discuss cell cycle and cell division, I told you, so one mother cell gives you 4 daughter cells at the end of meiotic division, much in the same way. So one primary spermatocyte undergoes meiotic division 1 and it forms your 2 secondary spermatocytes. So each of the secondary spermatocytes forms your spermatids. Right? Now these spermatids get converted into your sperms. So these are the sperms. So the development from the spermatids to the sperm, so this process is called as spermiogenesis. So again, genesis is formation. So when these sperms get released from the seminiferous tubules, or when they get released from the Sertoli cells, so that process is called as spermiation. Yes, clear? This is the process of your spermatogenesis. Clear? So far clear? So now let's quickly see what is the ploidy of each one of them. So spermatogonia is 2n. So again spermatogonia is 2n, primary spermatocyte is 2n, secondary spermatocytes, these are haploid, spermatids are haploid. So again sperms are haploid, because sperms are gametes, so always gametes are haploid, right? So far clear? So this is what we were reading in the previous lecture. So now we move forward to talk about the structure of your sperms. Right. So the process of spermatogenesis begins at the time of puberty, at the onset of adolescence development, okay. So how does it start? So just like that, your body will not start the process of spermatogenesis. So here you have the trigger and the stimulation of certain hormones. So which are the hormones? So increase in the GnRH. So what is your GnRH? So it is your gonadotropin releasing hormone. So that is done by the hypothalamus. So it acts on the anterior pituitary to release the gonadotropins, that is your LH and your FSH. So LH acts on the Leydig cells and it stimulates the synthesis of a particular androgen. One such androgen is your testosterone, which stimulates your spermatogenesis. FSH acts on the Sertoli cells and it also enhances the secretion of spermatogenesis. So how does the hormone stimulation happen? So your process of spermatogenesis it begins at the time of puberty. So how does it begin? So you have your hypothalamus. So the hypothalamus releases a hormone called as GnRH. So this stands for gonadotropin releasing hormone. So this hormone will act on the anterior pituitary. So anterior pituitary gland is called as adenohypophysis. Okay, so this will now release gonadotropins based on the instruction from your GnRH. So gonadotropins are a pair of hormones. One is LH, the other one is FSH. So LH stands for luteinizing hormone and FSH stands for follicle stimulating hormone, the hormones of the pituitary gland. So now this LH acts on the Leydig cells. So what are Leydig cells? So Leydig cells are the interstitial cells. Interstitial cells present in between the coiled seminiferous tubules. Alright. So LH cells, when it acts on the Leydig cells, they together produce the production of androgens. So one such androgen is your testosterone. So next is your FSH. Your FSH acts on the Sertoli cells, the one that nourishes the germ cells. So this FSH, which acts on the Sertoli cells, plus your testosterone, they stimulate the production or they stimulate the process of spermatogenesis. Fine? Yes. So first I told you the process of spermatogenesis, how it happens. I gave you recap. So now I am telling you how the hormonal action of it happens. Alright, clear? So structure of a sperm. So we're going to talk about the structure of the human sperm. So the human sperm has four structures. So So it has a head, it has a neck, it has a middle piece, and it has a tail. The entire sperm is surrounded by what is called as a plasma membrane. The head contains the haploid nuclei because the sperms have one nuclei, right? So the nucleus is present in the head of the sperm. Right? So the anterior portion, so on top of the head you have an anterior portion that is like a cap-like structure. So you wear your cap on your head, right? So similarly, just anterior to your head you have a cap-like structure. So that cap-like structure is called as acrosome. So what are the parts of a sperm? So you have the head, you have the neck piece, so you have the neck, the middle piece, and the tail. Right? So the entire structure of the sperm is covered by the plasma membrane. So anterior to, so the head has the haploid nucleus. Anterior to the head you have the cap-like structure, that is your acrosome. Right? So acrosomes, they contain enzymes that help in the fertilization of the ovum. So the middle piece has a lot of mitochondria, so which actually supports the sperm with the energy to move. Sperms are motile, right? Yeah, so this middle piece, which has a lot of mitochondria, produces energy, so which facilitates sperm motility, which is essential for fertilization. Alright, so here we have the structure of the sperm. So if you can see, so you have the head, so you have the neck region, you have the middle piece, and you have the tail. So the four parts are here. The head contains the haploid, so the head contains the nucleus. So inside the nucleus you have the chromosome material. The chromosome is haploid here. So anterior to the head you have a cap-like structure, so that is our acrosome. But if you see, the entire tail is surrounded by the plasma membrane. The middle piece has mitochondria, which provides energy source and it enhances sperm motility. Fine? Yes. So the human male ejaculates around 200 to 300 million sperms during coitus. So coitus is the sexual intercourse. So at that time, the human sperms that get ejaculated are 200 to 300 million. So why is that such a large number of sperms are produced? The reason is here. So out of this 200 to 300 million sperms, only 60% are of the normal shape and size. So not all are of the proper shape, which has the head, the acrosome, the neck piece, the middle, and the tail. right? So out of the 60, only 40 show proper motility, that they can travel towards the ampullary region in the female genital tract. So seminiferous tubules releases sperms that are transported by the accessory ducts. So who are the ducts? So you have the rete testis, so you have the vasa efferentia, you have the epididymis, and finally the vas deferens. Fine? Yes? So secretions from these, that is the epididymis, the vas deferens, seminal vesicle, prostrate. The seminal vesicle and prostrate are the glands of your male. So all of them help in the motility of the sperms. So the secretions of these glands, that is seminal plasma, plus the sperms together is called as the semen. So the functions of the male sex accessory ducts and the glands maintained by the testicular hormones, that is androgens. So these male accessory ducts and glands have a particular function, that is to enhance the motility of the sperms, and that is kept in control by the release of androgens. Alright, yes, did you understand? So why is that sperms are produced in large number? So during each of the coitus, at one sexual intercourse, at one coitus, you have the production of 200 to 300 million sperms. So why so? So why is that such a large number of sperms have to be produced? So the answer is, out of the 300 to 200 million, so only 60% are in proper shape. So out of the 60%, only 40% show motility, because they have to reach the ampullary isthmic junction of the fallopian duct in the female genital tract, right? So who helps in their movement are your accessory ducts. So which includes your rete testis, your vasa efferentia. So you have your epididymis. Then you have vas deferens. So these are the ducts, apart from which you have the glands like the seminal vesicle and the prostrate glands. So the secretions of all this help in the motility of the sperms. So all these secretions are called as seminal vesicles, seminal plasma plus the sperm together is called as the semen. So the function and the secretion of these axillary glands are kept in control by the regulation of androgens. Yes, understood? Clear? Yes? Alright, so with this we are done with the process of spermatogenesis. So now we would talk about oogenesis, but I will continue oogenesis in the next lecture. So oogenesis will be done in one lecture, so I don't want any interference. So give me 3 more lectures or probably 4 more lectures to be done with this. So one for oogenesis, one for menstrual cycle, so one for how implantation happens, fertilization and implantation, and the last part is how your fetal development parturition happens. The chapter will be done. So after this chapter will be done, so I would like to do one shot, or I would like to discuss previous year question. So that is your wish. So again a polling will be done, so please make sure you answer the polling. So whether we have to start with one shot or if you want me to teach another chapter, so the open it is open up to you. So you guys will tell me, so what are we going to start next with? Alright, yes. So I hope you have understood the process of spermatogenesis. So please start your preparation. So it is just 60 days, you could say, on an average. So any plus or minus will not make much of difference. So please be very diligent in your work. So start preparing, study well, have good time management. So though they are giving you 20 minutes extra, it does not mean you will sit all the 90 questions in biology for 1.5 hours. No. No, no, no. So 90 questions, 90 minutes is also too much of a time. So 90 questions, make sure you are done with 45 minutes. So that is what will do your best, alright. So time management is very important. So very soon we'll have a mentee, alright. So we'll continue in the next class, that's on Wednesday. So until then, this is me signing off. So take care, bye, and thank you.